Good day. My name is Abdulhafeez Babalola from University Sains, Malaysia. I'm excited to present my research titled A Highly Accurate Breed Random Forest Gradient Boosting Small Printing Frame Identification Algorithm Validated using Mycobacterium tuberculosis transcriptomes. Introduction. Random forest is an ensemble learning method that builds multiple decision trees and combines their predictions to enhance accurate performance, while gradient boosting sequentially trains the weak learners to minimize the loss function. Small operating frame is an operating frame with a length less than 100 amino acid, which can be located within the coding transcript of 5 untranslated region, the coding sequence, or 3 untranslated region. It can also be found within the non-coding RNAs. Polycitronic region refers to segments of genome where multiple genes are transcribed together, while intergenic region is a stretch of genome located between genes. Objectives. Development of a hybrid algorithm for the prediction of small proteins. Evaluate the performance of the algorithm. Validate the predicted small operating frame from Mycobacterium tuberculosis by analyzing transcriptome data. Identify and characterize the predicted small proteins. The methodology goes thus: compilation of our dataset from NCBI, which consisted of 46 bacterial genomes, and Mycobacterium tuberculosis for transcriptome dataset. Extraction of polycitronic and intergenic region of these bacterial genomes using GFFEX tools. Random forest and gradient boosting were introduced for the prediction. Different metrics were used for the performance evaluation, including accuracy, the sensitivity, specificity, area under curve, Matthew correction coefficient, including F1 scores. We predict small printing frame less than 100 codon. We further validate the predicted small printing frame by mapping to its transcript, where the transcript were identified from a transcriptome analysis. The data preprocessing of this transcriptome analysis were conducted using Trigonometric script. We map using Borowiller aligner. We introduced string type script for the transcript compilation and the expression estimation. The identified transcript and the predicted small printing frame were further conducted functional analysis using BLAST2Go and the Shiny GO for the identified transcript. Results. The performance evaluation of the predicted algorithm. The blue color is denoted for random forest, while the brown color is denoted for gradient boosting. The gray color indicated the hybrid algorithm. At individual prediction between random forest and gradient boosting, random forest has been able to achieve higher accuracy of 96% compared to gradient boosting, which achieved 93%. Random forest has been able to achieve higher performance at different metrics compared to random forest. While leveraging the strength of these two algorithms, making it an hybrid algorithm, the hybrid algorithm has been able to achieve higher accuracy at different metrics compared to the individual algorithm, while the hybrid algorithm has been able to achieve 99% compared to random forest and gradient boosting. The area under curve of random forest and gradient boosting, we're able to identify that random forest has been able to achieve 97%, while the gradient boosting has been able to achieve 94%. Leveraging the strengths, the hybrid algorithm area under curve has been able to achieve 99%. To further validate our predicted small proteins, we compare the predicted small proteins to its reference genome of Mycobacterium tuberculosis. While the purple color is indicated for the reference genome of Mycobacterium tuberculosis, and the green color is indicated as regions of predicted small proteins. The small protein frame predicted ranges, we are able to identify different ranges within the predicted small proteins of Mycobacterium tuberculosis, totaling into 557 regions. We further conducted functional annotations to provide insight into their functional significance, including the molecular functions, cellular components, biological processes, and the gene ontology levels of annotation. The identified transcripts were further performed functional enrichment of this identified transcript, including the biological process denoted in A, the cellular components, and the molecular functions denoted in C. To further validate the predicted small proteins, we compare to different species where we compare this analysis of Mycobacterium tuberculosis compared to its identified transcript. We identify total mass of 420, while 367 regions shows 100% similarity to query sequence, while the alignment of 90% above shown 403 alignment that have a similarity score above 90. This study predicted small proteins compared to previous study of Smith et al. 2022. The total alignment identified was 221, while 221 out of 221 matches show above 90% sequence identity. We further compare the predicted small proteins of Mycobacterium tuberculosis versus the pathogenic and non-pathogenic bacteria genomes within the 46 predicted small proteins. 95 small protein frame predicted pathogenic regions overlap with Mycobacterium tuberculosis, while 16 small protein frame predicted non-tuberculosis overlap with Mycobacterium tuberculosis. While the Mycobacterium tuberculosis were compared to other species of fungi, Trichophyton rubrum, while 5 regions with significant similarity were identified at a stringent e-value of 0.00001. Discussion and conclusion. The performance of the algorithm indicated that the hybrid algorithm achieved better performance with area under cover of 99% compared to individual algorithm. 5S7 predicted small protein frame regions encode small proteins in Mycobacterium tuberculosis less than 100 amino acid. The transcripts were accurately matched to predicted small protein frame, showing higher degree of similarity between them. The analysis shows that the predicted 557 of Mycobacterium tuberculosis align uniquely with the reference genome, with 88.82% having over 90% sequence similarity. The comparative analysis of the predicted Mycobacterium tuberculosis, including the previous study, the reference genome, and pathogenic and non-pathogenic bacteria within the 46 bacterial genomes, elucidate the conservation and potential roles of these small proteins in pathogenicity. The gene ontology analysis enriched the understanding of the biological significance of the predicted small proteins, shedding light on their diverse functions. Therefore, the study significantly advances computational bacterial genomes by providing a robust algorithm for identifying small proteins and provide valuable insights for future research on functions of small proteins in bacteria. Acknowledgement, references. Thank you.